This second part of cytology is going to address just the inside of the cell, and we're going to do it in a quick and easy, quite superficial form, just to confirm that we completely understand what's happening inside the cell. And I'm sure that you would remember it from a previous video that was addressing components of the plasma membrane that we reached this diagram as the stop point. So I promise that we're going to use it as the starting point for this section, which is about cytosol or cytoplasm. When we use the word just cytosol, we're referring to just a liquid part inside the cell, as opposed to using the term cytoplasm, which includes cytosol, the liquid part, with all the organelles that are found within the cell. Of course, with the exception of nucleus, that is going to be addressed in a third separate part of this mini-series about cytology. So let's take a look first and see what some of these filaments are interesting for us about. Basically, we refer to them as some kind of cytoskeleton, although they do not have exactly the same firmness as our bones would have. Maybe the term is slightly misleading, but there is really nothing better than to use this idea and to compare it to something that's quite tangible, as opposed to just look at submicroscopic structures. Microfilaments are first group of proteins, which you can see being practically lined up next to the cellular membrane, and of course, seeing them as penetrating into any kind of projections that cell on one of its surfaces would have. In this case, it's a series of microvilli that are increasing the absorptive surface of the cell. Intermediate filaments are running practically without any particular arrangement throughout the cell, from left to right, front to back, bottom to the top, or the other way around. And they really more internally regulate what the cell is gonna look like when all the organelles are properly populating the inside of the cell in cytoplasm. So they would prevent any kind of excessive displacement of, let's say, nucleus finding itself next to one of these microvilli, or endoplasmic reticulum separating and going too far away from the nucleus. Practically, it's just internal furnishing of the cell, which is arranged and guaranteed by these intermediate filaments. Finally, we're going to have some components of the cells that are made by microtubules. As you can see, really, size of microfilament, intermediate size filament, they both appear to be solid, where proteins building microtubules actually are creating some form of tubular spaces that exist within these organelles. And in this case, it is going to be illustrated through organelle that is called the centrosome, which is essentially very important for the cell while it undergoes its cellular division or mitosis. If we take a closer look at the centrosome, you can see that it really looks like being made of two parts, two tubular structures, which intersect each other at a 90 degrees angle, and forming the walls of microtubules are a set of these three longitudinally arranged proteins that are creating a bit of centrosome and its wall. Basically, we have microtubules coming in triplets, and this kind of purple bluish cloud which surrounds the centriole is called the pericentriolar material. That's going to be used for the cell during its cellular division to bring changes and to form mitotic spindle, which at the end will be used to separate chromosomes and to pull them towards the opposite poles of the cell. Lower picture is just 76 times magnification electronic microscope, identifying centrosome just the way it is. This is artistic redemption, and of course, is a little bit easier for us to observe and to try to understand what's going on. Similar arrangement of proteins in a set of three, creating some form of other microtubule, is going to be found not necessarily within the centrosome, but rather within part of the cell, which is called cilium or flagellum. In first part of these videos about cell, we introduced the idea of cells, not always, but some of them exhibiting cilia or flagella, which are contributing to mobility of the cell. Of course, most of the cells are immobile, whereas male reproductive cells having flagellum, which keeps actively moving, male reproductive cells are those ones that exhibit ability to move. If we are using the term cilium, then it's also like built by on the basis of microtubules. However, cilia are much shorter than a flagella, and cilia, number of different cells would have them, which would also exhibit active movement, but not of the entire cell, but rather just of the ciliated part of the cellular membrane. Good example are cells of the upper respiratory tract, which are loaded with cilia, because as we breathe, inhale and exhale, possibly they would trap some contaminating particles that would from time to time need to be eliminated. So with the beating of the cilia one way, fast, and recovery being relatively slow, they actually move the particles on the surface of the cell, which is ciliated. Interesting and very important small organelle is known as the ribosome. It is found in the vicinity of nucleus. Usually most of them are seen attached to endoplasmic reticulum, to its rough part, and it's the ribosomes that are making it looking rough, because millions of ribosomes are attached to that particular section of endoplasmic reticulum. Individual ribosome, when enlarged and when illustrated, consists of two units, the large and the small subunit, which need to come together to form complete functional ribosome. The most important, the principal role that ribosomes would have is to be part of protein synthesis and to be able to read the code that is embedded into a molecule of messenger RNA, going from 3 codons to another group of 3 codons, identifying individual amino acids which need to be added into chain in order to bring the protein structure. We're going to see them in a section about nucleus, and we'll understand a little bit better what they do and how they help us synthesize proteins and to make form of chain of amino acids. We already mentioned endoplasmic reticulum being close to the nucleus of a cell, and the rough part, the rough endoplasmic reticulum, actually looks like rough on its surface because of literally thousands, if not millions, of ribosomes that are attached onto its surface. Endoplasmic reticulum, a rough part, is found directly in the form of concentrated little flattened sacs that are surrounding the entire nucleus. And then after ribosomes have populated its surface, the rest of it, which we can see more peripherally located in a cell, becomes the smooth endoplasmic reticulum. Clearly, smoothness comes from lack of ribosomes from its surface. However, both smooth and rough endoplasmic reticulum are important because of their ability to participate in the synthesis. The rough endoplasmic reticulum is more important for synthesis of proteins, while synthesis of lipid-based molecules such as estrogens or progesterones or testosterone, essentially lipid-based molecules are synthesized within the smooth endoplasmic reticulum, after which they would be allowed to get transportation vesicle and to be exported from a cell capable of synthesizing those precious content. Another interesting organelle, shape-wise quite comparable to endoplasmic reticulum, is also made with a series of flattened parallel running sacs, but this time we're talking about Golgi apparatus. If a protein is synthesized from rough endoplasmic reticulum, those transportation vesicles, which detach from endoplasmic reticulum, are now merging with the flattened sacs, so-called cis face, the face or surface oriented towards nucleus, and then they blend with the membrane of Golgi apparatus, releasing the content that they have transported. Within the endoplasmic reticulum, synthesis took place, but as we know, proteins are still subject to some kind of modification. Just observing the chain of amino acid is a primary structure of the protein. And then if we need to fold it or bend it to get into three-dimensional shape, those changes, tertiary, secondary, and finally quaternary structure of a protein actually takes place within the Golgi apparatus. Golgi apparatus, with a finalized product, also prepares its own transportation vesicles, which pinch off from the surface of Golgi apparatus, and they drift away towards cellular membrane, carrying a substance that will likely be exported from the cell, and surface from which those transportation bubbles are released is away from the nucleus, and we call it the trans face or trans surface of the Golgi apparatus. On this slide, we can see a little bit better, a little bit closer as what's happening. This is rough endoplasmic reticulum, and we see here synthesized protein within RER getting into its own transport vesicle, which pinches itself off and moves away from endoplasmic reticulum. It travels short distance before it lands on the surface of the Golgi apparatus, and the protein, which is now very simple, looking just like a chain of amino acids, enters into cistern or tubule of Golgi apparatus, which slowly and gradually starts making needed changes and transformation. So when the protein is finally formed in its secondary or tertiary or quaternary form, basically the ready product is shifted to the end of Golgi apparatus, whether it is on its trans side, where it's going to be exported in the form of secretory vesicle, or the protein might be recycled once again and to let it have another pass through the Golgi apparatus if additional changes are required. So if we take a look at these three secretory or transportation vesicles that are to be formed, we can see that if we follow just the red colored one, it would contain a product which essentially would be lots of different enzymes that will be transported by appropriate vesicle to the lysosome. So it'll replenish its own enzymes, where this purplish colored transportation vesicle carrying some interesting proteins are going to be practically proteins that will be rebuilt and left in place, being those proteins that are part of the cellular membrane, part of it sticking out into extracellular surface, part of it sticking and protruding into intracellular surface. And finally, the blue colored one, the secretory vesicle, will travel towards cellular membrane, will merge with it, and of course then it will open itself up and release the content which has been transported from Golgi apparatus to extracellular fluid. That is the process being called exocytosis, elimination of substances or structures from inside the cell. Opposite part was endocytosis that we discussed in the first part of this video. Finally, the lysozyme, which is another important and interesting organelle, which comes in multiples scattered throughout the cells. Practically, we know that lysosome contains a number of different digestive enzymes. So when the lysosome merges with a transportation vesicle, basically it will release its enzymatic content that'll start breaking down the substance that has been imported. It'll make it either harmless or it would be completely digested and converted into energy. And its byproducts will be either reused as a recyclable material, amino acids, to be used eventually by ribosomes, or if byproducts are simply of no further use, they will be eliminated from the cell. Finally, we become now more informed about different structures, and I believe this is the last one on this slideshow. It is another important organelle. It's mitochondria, so-called the powerhouses of cells. As we can see, they're a very complex structure. They have outer mitochondrial membrane, which simply keeps the physical boundaries, but inner mitochondrial membrane is thrown into multiple folds that are called mitochondrial cristae. They also have plenty of ribosomes that are attached to it, but as well as plenty of different enzymes that will help us at least understand single statements saying that ribosomes are structures or organelles within a cell that generate adenosine triphosphate. So for that reason, they're seen as the powerhouses of the cell. This slide is going to be our end slide for this section, second section of video about cell. And we're going to use it as beginning for the last third part, where we have to say a few words about nucleus and its content.